In this tutorial we will discuss the Data Import and Project Creation module. ShIVA offers 3 ways to import data. The first method is to import files from the analysis of the sequencing data by the Cell Ranger software, developed by 10x Genomics. The second method consists of loading a binary object in RDS format, produced with the R software, using the SURIT package. The third method allows to load a project saved from ShIVA and to recover an analysis already carried out. We will start with the first method, that is loading files from the analysis of the Cell Ranger software from the company 10x Genomics. We first need to load a first set of files which can contain either only RNA-related data, or multiplex data, for example, RNA and HTO, and potentially also CITE-seq antibody captures. In this example we will load RNA-only data. By clicking on the Browse button, we see a directory in which the 3 files provided by the Cell Ranger software are present, that is the file containing the list of cell barcodes, the file containing the list of features, in this case detected genes, and the file containing the count matrix. We have to select all 3 files at the same time by shifting and clicking the mouse, and then ask ShIVA to open them. ShIVA then loads the data. Once the data is loaded, we see that ShIVA presents us with a table with information about the loaded data. As we have only loaded RNA data, there is only one line in the table that shows us the gene expression, with more than 10,000 cells and some 33,000 genes. It is possible in a second step, if we have only loaded RNA data, to also load data containing HTO, or data containing ADT CITE-seq antibody captures. In this example we will also load HTO data. To do this, as before, we click on the Browse button and navigate to the directory containing the data files, here produced by the CITE-seq count software. The data is then loaded into ShIVA. A new row of information appears in the table, stating that this is HTO data, the number of cells and the number of features, in this case 4, as there are 4 samples. The Venn diagram on the right allows us to identify the intersection of the barcodes between the 2 datasets, which here is perfect. All the barcodes found in the RNA data are found in the HTO dataset. It is possible when we load HTO data, to edit the names of the associated samples. Here we see 4 names that are provided by the data. We see that the names are long, and carry the HTO barcode. ShIVA has an automatic way to remove these HTO tags to make the names more readable. All you have to do is click on the Remove Barcodes button. The names are then simplified and we can continue loading. If the CITE-seq data is available, we can also load it via the third load box. Once the data is loaded, we need to create the project. To do this, we will give the project a name. This name must be explicit so that it can be easily identified later on. As our data is follicular lymphoma data, we will call the project FL Tutorial. You must then choose the species associated with your data. The choice is limited but only serves to preload some useful files later. Next, we need to choose the method for normalizing the data. ShIVA offers 2 popular methods, log normalization and SC transform. Here we choose the log normalization method. We then have to choose 2 parameters. The first one asks to enter the minimum number of cells in which a gene must be detected to be retained for further analysis. If we enter 3, only genes that are expressed in at least 3 cells will be retained. Genes that are expressed in less than 3 cells will be discarded. You can vary this number. Similarly, the second parameter asks you to enter the minimum number of genes that a cell must express to be retained. If we enter 200, only the cells that express at least 200 genes will be kept, the others will be eliminated. This initial filtering is coarse and can be refined in the quality control module. Once the data has been entered, we can click on the Create Project button to launch the creation of the project. Once the project has been created, the number of cells loaded, the number of genes and the number of HTOs appear at the top of the interface. As you can see, the number of cells is slightly lower than the number of cells initially loaded. And the same is true for the number of features. This is an effect of the filters we placed when creating the project. A second method of loading data that ShIVA offers is to load a file from the analysis through the SURIT package of the R language. A bioinformatician can take your data, produce a file in RDS format in R via the SURIT package, and provide it to you for loading into ShIVA. Here we will browse the file directory for the prepared RDS file and load that file. Once the data has been loaded, we can see that the RNA data and HTO data information, the number of cells and the number of features appear directly. We then have to enter the name of the project and the species. The project can then be created. The last method available under ShIVA to load data consists in loading a file resulting from the export of the analysis by ShIVA. There are 2 possible methods. The first is to use a project that is automatically saved by ShIVA. Indeed, when you perform your analysis under ShIVA, automatic backups are made on the server where ShIVA is installed. You can then easily, by date and project name, find the project and load it. Another solution is to import an RDS file, which has been exported by ShIVA. This export can be done via the red Download ShIVA Project button. When this file is loaded, the project is then directly created and appears on ShIVA. Once a project has been created, it is possible to start the analysis by going either to the HTO demultiplexing module, if your data contains HTO, or directly to the quality control module, if you have no HTO in your data. The successive modules of the analysis are shown in individual videos.